Okay, so in the early stages of the secretory pathway, you have synthesis of proteins in the rough ER, and once the proteins are synthesized, they uh, they could either be soluble cargo proteins such as this one, or they could be embedded in the membrane such as these membrane cargo. And usually, once the proteins are ready for transport to the Golgi, they form these. They are enclosed in these transport vesicles called the COP2 vesicle. And so you can see here is the vesicle forming, and eventually it'll pinch off, and this green protein coat will shed in step 2, and you'll have these membrane proteins exposed to the outer surface. Binding occurs to the cis-Golgi network through these snare proteins that pair up and form coiled coils. And then once the membrane is fused with the cis-Golgi, some of these proteins here shown in blue and orange could be recycled back to the rough ER through a retrograde movement through these COP1 vesicles, which have a different protein coat, purple versus green. And these COP1 vesicles will transport some of the cis-Golgi network proteins back to the ER, and they have their own snare pair. Now, one of the first steps in the formation during budding is the formation of this complex here, which is a tetramer. So the first protein that is involved in initiating the formation of these vesicles that will bud off of the rough ER is SR1. Now SR1 will bind to SCC23 and SEC24, and once they form this tetramer on the ER membrane, some proteins in the membrane which have a cytosolic facing segment, if they make favorable contact with SEC24, they could be transported as cargo protein to the cis-Golgi. And also, some of the proteins that are formed in the rough ER that are involved with helping other proteins fold, sometimes they're accidentally transported from the rough ER to the cis-Golgi, and the way that they're returned back to the rough ER to maintain a steady population of this protein, you have this KDEL receptor, which binds to a conserved segment of these proteins shown in purple or pink here. And so once the protein is delivered to the cis-Golgi, the KDEL receptor will recognize it. They bind to each other, and they're incorporated into these COP1 vesicles, which are transported to the ER. And once they bind to the ER, or once the COP1 vesicle fuses to the ER, it releases the protein into the ER, and this release occurs through a change in pH. So you can see here for the rough ER, it's at lower proton concentration, which means it's at a higher pH. Whereas for the cis-Golgi network, it's at a lower pH. And so at a higher pH, this binding is not favorable, and so they dissociate. Also, some of the steps that form glycoproteins in the different compartments of the Golgi apparatus, such as the cis, medial, and trans, are summarized by this diagram, which shows that in the cis compartment of the Golgi, these glycoproteins that have 8 manos, shown here in blue, are trimmed by a enzyme into a 5 manos unit. And in the medial compartment, you have the addition of 3 N-acetylglucosamine units, and also the removal of 2 additional manos. And also on the very first N-acetylglucosamine in red, you have an insertion of a fructose molecule. And then in the trans compartment, you have every N-acetylglucosamine in red is going to be attached to a galactose in orange, and an N-acetylnuramic acid, after which they exit the trans compartment and go to their proper destination. And then there's an electron micrograph showing a image of the early stages of the secretory pathway of a pancreatic and cinder cell. And so you can see here's the ER, the rough ER, in the bottom here, and you have the formation of these COP2 transport vesicles occurring through the budding process. So they bud off of the ER and they move towards the cis-Golgi. And so in the cis-Golgi, you have the fusion of a lot of different COP2 vesicles. And once the vesicles fuse to the cis-Golgi network, they mature into the medial and eventually the trans, after which they bud off into their own secretory vesicles that will go either to the plasma membrane or an endosome or lysosome. And one of the ideas here is that so one of the ideas here also in the early stages of the secretory pathway is that cargo protein they do not bud off and go to the next compartment of the cis-Golgi. Instead, these different compartments, they mature into their next stage. So this cis will eventually mature into a medial and then into a trans Golgi compartment, and from here they bud off and go into their different directions. And so proof of that is from these type of fluorescence experiments, in which you have here 2 different proteins here in red and green. And so VRG4 is a protein involved in the early Golgi process, and it is fluorescing in green. And so you see, yeah, the green is for the early stage of the process, and then another protein involved in the Golgi network called SEC7. It is fused to a red fluorescent tag. And so you see, if we focus on one compartment of the Golgi network, you see that initially it is associated with the early stage protein VRG4 in green, and over time it loses that green fluorescence and also gains more red, which is the SEC7 used in the later stages of the Golgi processing. And so you can see it transitions to red, and so this shows that one compartment had it lost its initial protein and gained a protein involved in a later stage, and this occurs through retrograde vesicle transport, in which if we see this this diagram here, proteins involved in the later stages of the processing in the Golgi transmembrane, through the retrograde system, they will be brought back down to an earlier stage compartment, and then because of that, this compartment will mature into a later stage. And at the same time that this compartment would be gaining enzymes from the trans compartment, it'll be losing its own medial-related enzymes to the cis compartment, since COP1 vesicles will move proteins retrograde to an earlier stage. And so that's the idea of the cis Golgi compartments, is that they mature over time, and cargo proteins don't necessarily move from one compartment of the Golgi network to another. They stay within that one compartment that they initially fused into, but they mature over time through the changing species of enzymes dissolved inside that compartment through the retrograde system.